and it's a pleasure this evening to present the Trainee Science Communication Award. So this was established in 2020, and it recognizes the exceptional outreach and mentoring efforts of a student or postdoc member of the SDB. This year, Victoria Deneke, postdoc in the lab of Andrea Pauli at the Research Institute of Molecular Pathology, is winner of the Trainee Science Communication Award. Now, Victoria is an absolute rising star. She's won so many awards, such as the Harold Weintraub Graduate Student Award in 2020, an HFSP Program Postdoctoral Fellowship, and an HHMI International Student Fellowship. But today she's receiving this award for her many outreach initiatives and mentoring of undergraduate students that we are about to hear about. So it's my pleasure to introduce Victoria. Thank you.
So throughout this meeting, we've heard about fascinating discoveries in developmental biology, ranging from the remarkable organization within developing systems, such as these cortical waves in barnacle embryos from Aaron Jesuit, to the power of organoid systems, such as these somite-like structures that form in human embryonic organoids from Mikia bisuya. And a common thread that underlies much of developmental biology is the ability of organisms and living systems to undergo transitions. So today I'd like to share with you two topics that I profoundly care about. What brings them together is the fact that they are both at the heart of a transition. So in the first part of my talk, I will share with you my postdoctoral work in the group of Andrea Pauli, where I have been studying a fundamental transition that results in the activation of developmental programs, and that is the process of fertilization. And in the second part of my talk, I will share with you a transition that I envision for academia and how I see that outreach and science communication will play a key role in this transition. So personally, I find the process of fertilization is one of the most remarkable biological processes, whereby the sperm, one of the smallest cells in sexually reproducing organisms, binds and fuses to one of the largest cells in animals, the egg. This is a movie of fertilization in starfish. And upon sperm-egg fusion, there's an increase in calcium levels in the cytoplasm, which is depicted by a yellow flash. And this increase in calcium marks one of the first activation cues that initiate the process of embryonic development. So what do we know so far about how this process is regulated at a molecular level? In mammals, so far, 7 sperm proteins depicted here in teal and 2 egg proteins depicted in orange have been shown to be essential for spermic binding and fusion in mammals. So in other words, knockout mice for each one of these genes show severe or complete sterility, and the fertilization defect is at the very last step of binding and fusion. Importantly, there has been one protein pair that has been shown to interact between sperm and egg, and that is Izumo and Juno. So what are we missing? If we zoom out and think a bit more broadly about the process of cell fusion, it involves rearrangements of plasma membranes through a series of conserved steps. First, the close apposition of the 2 plasma membranes, followed by the formation of a fusion intermediate during which the outer lipids mix, and then the opening and expansion of a fusion pore. However, cell fusion, as you can imagine, is a thermodynamically very unfavorable process. So there's a need for specialized proteins called fusogens in order to mediate this process. So if we look at other examples of cell fusion in biology, like viral entry, placenta formation, muscle development, and regeneration, we find that there are specialized proteins that can drive fusion in these systems. For example, in viral entry, you may be familiarized with this protein, spike, which is present on SARS-CoV-2 and drives the viral fusion to target cells. Interestingly, syncytins, which drive the fusion of trophoblast cells, were endogenized from retroviruses and share remarkable structural homology to viral fusogens such as spike. Myoblast fusion is known to be mediated by two fusogens called myomaker and myomerger. And finally, there is a eukaryotic fusogen that has been shown to mediate gamete fusion in plants, algae, and protists called HAP2 or GCS1. Interestingly, HAP2 shows similarities to other viral fusogens like that of tick-borne encephalitis virus. However, even though HAP2 has been found to be broadly present across eukaryotic species, there is no HAP2 homologue in vertebrates. So this leads to a very interesting conundrum. And given that the currently identified proteins in vertebrate gamete fusion don't share any structural homology to known fusogens and don't have any regions that resemble a fusion peptide, it is currently unclear how these set of proteins mediate gamete fusion, or whether we are still missing factors in this picture. So we are using zebrafish as a model organism to study fertilization due to several advantages, including the fact that they undergo external fertilization. We can also collect a large number of gametes for biochemical studies, and it is amenable for imaging. The eggs of many fish and insects have a funnel-like structure that I have depicted here called the micropyle, which is the only entry site for sperm, which makes it very convenient for live imaging. I will now show you a movie of what fertilization looks like in zebrafish. So here, you can see the egg, and you can see the funnel-like structure, the micropyle. And once I play this movie, you will see the sperm accumulating in the micropyle. They're coming in. Each one of these dots is a sperm. And you will see these calcium transients in teal in the egg. And what you will notice is that after a few of these transients, the top egg coat lifts off, preventing any further sperm from entering. And only one sperm managed to get in and will initiate embryonic development. And I find this truly, truly beautiful and fascinating. So what do we know about the molecular regulators of fertilization in zebrafish? So compared to the mammalian system, several of the factors that are essential in mammals are conserved in zebrafish, and I have highlighted them here in teal. Surprisingly, though, there are several other factors that are not present in fish, such as the isomo interaction partner, Juno. This makes zebrafish a very interesting and perhaps simpler system to complement mammalian studies that can allow us to study the function of these conserved proteins, as well as the diversity of reproductive strategies within vertebrates. In addition, our lab discovered a protein called bouncer that is expressed on the surface of eggs, and it's required for fertilization in zebrafish. Interestingly enough, the mammalian ortholog is not expressed on the egg, but rather on the sperm, and it is important for the penetration of the sperm of the egg coat by the sperm. And this is an event that happens upstream of plasma membrane binding and fusion. So in fish, bouncer shows species specificity between zebrafish and medaka. In other words, by expressing the medaka bouncer on zebrafish eggs, you can fertilize zebrafish eggs with medaka sperm, which I find truly fascinating. So as a postdoc, I have been involved in studying two of these conserved factors within vertebrates, SPACA6 and DCST1 and 2. And what we have found was that they are essential for fertilization in zebrafish as well as in mammals. And even though these factors are not required for sperm-egg binding in zebrafish, they are required for binding in zebrafish, they are not required for binding in mouse, showing that there's already differences there between these two vertebrates. And this suggests that perhaps these proteins may have evolved species-specific functions or species-specific protein-protein interactions. Alternatively, the mammalian model could have different proteins present on the sperm that then compensate for this binding. So finally, in both systems, there is evidence that these factors may be indirectly or directly regulating each other. So we are very excited to uncover the basis of this regulation and how these factors could be coming together to regulate gamete fusion in vertebrates. So I would quickly like to highlight that this BACA-6 study was led by 2 talented undergraduate and master's students whom I had the privilege to supervise and mentor. And this publication really epitomized the role of mentoring, science education, and teamwork, which are some of the fundamental pillars that I will focus on during the second part of my talk. And finally, we have identified a new conserved factor expressed in sperm that has not been characterized before in any system. And we have preliminary evidence that it is required for fertilization both in zebrafish and in mice. So I really hope that I will be able to share with you in next year's SDB meeting our findings from this work. So just like fertilization is one of the key steps that enables embryonic development, I think that there are 3 pillars that will be key to focus on as we improve the academic enterprise. And these are education and mentoring, science communication, and community enrichment. So I will share with you 3 initiatives that I think embody and capture the essence of these pillars. I wanted to first give a very brief overview of my trajectory, since this has been a major driver and motivator for a lot of my outreach work. I was born and raised in a small country called El Salvador, which is situated in Central America. And from there, because I had access to good education in El Salvador, I was able to receive a scholarship to attend the University of Notre Dame for my undergraduate studies. After that, I moved to Durham to pursue my PhD at Duke University, and I currently have moved across the Atlantic Ocean to IMP Vienna for my postdoc. And after living in the US for a few years, I realized that I was in a very unique position to contribute to science education in my home country of El Salvador. And I had received an amazing science education in world-renowned institutions. So I started to contact local universities back home to see how I could contribute to developing STEM in my home country. And so I partnered up with a local university in El Salvador who had been running a summer STEM program for middle school girls. And the goal of this program is to introduce young girls to different topics in STEM and create a support network. Currently, most STEM programs in El Salvador have a strong male bias. So this is key in El Salvador. So I helped design workshops that would come in, and I would come in during the summertime throughout my PhD to teach these workshops and support this program. And on the right, there's a photo of one of these workshops where we were introducing students to the concept of pulleys in physics. And what I love about this program is that it embodies 3 key aspects that I think are essential to foster within our scientific community. First, educating the next generation of scientists. Secondly, communicating science at different levels. And thirdly, creating community within our science institutes to support people from all walks of life. So the second story I would like to share with you started out at a conference. I was attending ASCB in December 2018, and I was walking through the poster session, and I saw a stand, and that there were several people gathered around, and I saw this do-it-yourself microscope. And everybody was looking through this, and at this stand was Bob Goldstein from UNC Chapel Hill, who was sharing his initiative to teach local educators and teachers from across North Carolina how to build these low-cost microscopes as a tool to teach science. And I was so fascinated by these microscopes that I asked Bob what he thought of the idea of taking this workshop to developing countries such as El Salvador. Bob was super happy to have me join for one of his workshops to learn how to make these low-cost microscopes and to be able to give a workshop back home. And here's a video of what that workshop looked like in El Salvador. So with the help of Bob, I translated all the material and organized the workshop with around 50 teachers and students from across the country. And we targeted teachers with the aim that as educators, they could amplify this knowledge in their classrooms. And here's just a very short clip of everyone assembling their microscopes and being very excited. And here are some of the things that they saw under those microscopes. So very simple things such as salt crystals and torn paper, or red onion, for example, looks fascinating under these $10 microscopes. And one of the most rewarding experiences was to see the faces of teachers and students seeing a leaf up close for the first time and just being totally blown away by the beauty of biology. And so what I learned from this initiative is that you don't need a lot of money to show people the beauty of science and to spark that sense of curiosity and discovery for our natural world. And so the final story I would like to share with you is that of a fellowship that we started together with my postdoc advisor, Andy Pauli. So coming from a developing country, but having had access to education, made me realize that a lot of success is often based on having access to education and resources to do research. And throughout my outreach work in El Salvador, I knew that there was a lot of talent, but these students would likely never have the opportunity to be in an environment where they can learn how to be scientists. So we decided to do a very small yet life-changing experiment. We interviewed undergraduate students from El Salvador and recruited a very talented Salvadoran to come do science with us for the summer. Eduardo would come into lab every day with a huge smile on his face, and he was so excited about everything we did. I remember the first time we cloned something and we were doing gel extractions, and he just said, "This is so cool." And just seeing his enthusiasm was so contagious, and it reminded me of the privilege that each and every one of us has to be doing science every day and to be witnessing these moments of discovery every day in our own labs. And so not only was his energy contagious, but I also learned a lot about how science is done in El Salvador. He told me that to run DNA gels, they have to build the electrophoresis chambers from scratch. And these kinds of stories made me truly appreciate the resources that we have in our labs and have a better understanding of the challenges that scientists in developing countries face. So after 3 months in the lab, Eduardo gave a phenomenal lab meeting and is now back in El Salvador, completing his last year of his undergraduate studies. Thanks to this experience, he is eager to apply to a master's program and eventually a PhD program to become a scientist one day. Not only that, he has taken a lot of the knowledge that he learned from the lab and started a journal club with his university classmates, and he is also setting up a molecular biology club in his university. So to summarize, these experiences really represent what science is about for me personally. It's not only about discovering and understanding the world that is around us, but also about mentoring the next generation of scientists, about learning how to communicate our findings, and about creating a community that is nourishing and that is inclusive to everyone. And I hope that just as fertilization sparks development, we can foster these pillars to bring about the ideal academia we all envision. So I cannot finish this talk without thanking my nominators, mentors, and mentees. And first and foremost, Andy Pauli and Stefano D'Italia, my postdoc and PhD supervisors who nominated me for this award and who have supported me with this outreach work that oftentimes took time away from the bench. And so for that, I really have valued their mentorship. They have taught me how to be a scientist. Not only that, but how to be an integral scientist. Secondly, I would like to thank Bob Goldstein for all the help for the do-it-yourself microscopes. He is happy to collaborate with people from across the world. So I encourage you all to reach out to him if this is of interest to you to impart in your local communities. I would also like to thank my high school mentor, Amadeo Cortez, who also helped me with the material with the outreach activities in El Salvador. Also, I didn't have time to share this story, but I would like to really thank my first research mentor, Dr. Karen Calden-Dahl at the University of Notre Dame, who believed in me. She gave me my first research experience, and at the time, I was a chemical engineering student with no biology background and no research background, and yet she believed in me and hired me in her lab. And finally, I would like to thank my mentees, Miriam, Anna, and Eduardo, who have really changed me as a person and as a mentor and have really made me grow. And I'm very inspired by them and the work that they're currently doing. So I would like to thank the SDB for this award. The SDB is paving the way to transform academia, as evidenced by the inauguration of this award, and also as evidenced by the Choose Development program. I think these are two amazing initiatives, and I am really honored to join the legacy of previous SDB Science Communication awardees, Cagney Coomer, who founded the nonprofit organization Nerd Squad, which aims to promote science and community in Kentucky and beyond. And I would also like to highlight Julia Dirk, who founded Clear Direction Mentoring, which provides mentorship to underrepresented minorities. Please follow their work. They are doing amazing things to transform our scientific community. So in the spirit of the SDB conference and the SDB community, I would love to start a conversation of how you see the future of academia and what you think are key steps that will facilitate that transition. So therefore, I invite you to tweet your ideas, and even if you don't have Twitter, come up to any one of us and we can tweet for you. And so you can tweet your ideas by using the hashtag #MyDreamForAcademia and hashtag 22SDB. And I'm really looking forward to read what everyone has to say, the main ideas that keep popping up, and also brainstorming ways to how to give life to each one of these ideas. So thank you once again for this recognition and for your attention.